That was terrific. And last is DJ Pan, who I guess I can say now is at UT Southwestern. So if you plan on working in DJ's lab, you'll be heading more south now. DJ has been a pioneer in the Hippo pathway, and he's going to discuss his work today in Hippo signaling and growth control and beyond.
I'd like to start by thanking the organizers for inviting me here, and I also thanking you for sticking around for the last talk of the session. So, how do I start here? I'm a PC person. All right. So the work on the Hippo pathway can be traced back to the long-standing interest by developmental biologists to try to understand the fundamental question in developmental biology, which is how tissues grow to their characteristic size. And as geneticists, we can take a forward genetic approach, which is simply to take any model organism and simply do random mutagenesis and look for genes which result in an aberration of organ size. And this approach has been particularly successful in fruit fly, where in the last several decades, people have been able to identify different kinds of overgrowth mutants, or so-called tumor suppressor mutants. And these mutants can be generally put into two categories, either those which result in a neoplastic phenotype, which we heard about in a beautiful talk last night from the Larry Sander memorial talk. But also there's a second class of overgrowth mutant called hyperplastic, in which the epithelial and apical basal polarity is maintained. And among these hyperplastic class, and of course there are those which need to increase in cell size and those need to increase cell number without changing cell size. It turns out, in hindsight, actually almost most or perhaps even all of these genes which belong to this group of hyperplastic class and turn out to either encode a core component or regulators of the Hippo pathway. So the Hippo pathway was really defined by 4 tumor suppressor genes, which were discovered by a number of labs. And these 4 tumor suppressor genes encode protein kinase cascade, in which these 4 tumor suppressors, Hippo, Sourdough, and Warts and Mats, and work together in a kinase cascade, which really functions through the phosphorylation of a key transcriptional co-activator called URKey. And phosphorylation of URKey actually prevents its function as a nuclear activator. And then in the absence of this phosphorylation, URKey can engage its partner scallops to activate genes which normally would promote growth. So URKey is really an oncogene, while these upstream tumor suppressors, of course, they suppress growth. The work actually in the last few years really demonstrated that the Hippo pathway is highly conserved between flies and mammal. So now we take it for granted, but it really I think it speaks to this this long-standing theme in fly research, where this pathway is discovered in fly and now turn out to be conserved in mammals, and just Hippo is just one of those examples. And so, for example, not only like in fly, if you actually activate this oncogene URKey, you result in a very severe overgrowth phenotype. But in mammals, similar phenotype can be observed if you take the mammalian counter of URKey and simply overexpress in a mammalian organ, such as the liver, and you also result in tremendous overgrowth. Studies in the last few years from many labs have now uncovered this general principle in which the Hippo kinase cascade from Hippo all the way to the transcription factor really function as a fixed biochemical module. And this biochemical module can be regulated by multiple upstream inputs. And these include genes involved in cell adhesion and intracellular kinases and the phosphatase, and proteins involved in cell-cell interaction, and actually even proteins involved in apical basal polarity, which actually initially was thought to be involved in the belong to the neoplastic class of tumor suppressor. So even all these protein tumor suppressors have now been somehow linked to this biochemical module that I just spoke about, from Hippo all the way to the Yorkie. But I believe many of the biochemical details of how these tumor suppressors are linked to the Hippo kinase cascade need to be worked out in future studies. And what is also very interesting from a recent study is that not only these tumor suppressors, but also even the actin cytoskeleton, and more recently, and the spectrum cytoskeleton are also somehow linked to the Hippo signaling pathway. And so the general theme is that the Hippo pathway is really a biochemical module that can be regulated by many different upstream inputs, such as cell contact, cell polarity, and cytoskeleton tension. All right? And so besides this idea that the Hippo kinase cascade functions as a biochemical module to receive multiple inputs, another theme that really emerged from the study of the Hippo pathway is that the Hippo pathway can be used in many diverse biological processes beyond growth control. While many of us started our work on the pathway because of our interest in growth control, and studying in the last few years have really expanded the role of the Hippo pathway into many developmental contexts which have nothing to do with growth control. And actually one of the most striking and beautiful examples of that is in early mammalian embryo, and which actually this is the very first cell fate determination event in a mammalian embryo, which is the specification of so-called inner cell mass versus the trophic ectoderm cell. And so this is bipartite cell fate determination event is really dictated by the level of Hippo signaling. So in the absence of Hippo signaling, these inner cell mass cell would differentiate as trophic ectoderm cells, and they turn on markers which are normally restricted to these trophic ectoderms. And there are many other examples as well, which I don't have time to talk about. But so really, now we know that the Hippo pathway not only receives multiple upstream inputs and from a molecular point of view, but also biologically, they play diverse roles in development and physiology. So today I'd like to share with you a recent story from our lab, which really speaks to both of the two themes, which is different inputs into the pathway versus as well as additional function of the Hippo pathway. And so really the story started, was really spearheaded by a very talented postdoc fellow in my lab, Bo Liu. So Bo actually came to my lab with a very strong background in virology. So he was actually studying how virus infect insects as a graduate student. So when he came to my lab, he wanted to study whether the Hippo pathway is involved in adult physiology. And so for the relevance of this talk, what he discovered is that actually the Hippo pathway is intimately linked to the innate immune response in flies. So before I go into the detail of his work, I really should introduce you to the fly innate immunity pathway. And there are two major pathways which were discovered in beautiful work done by pioneers in Drosophila. And so basically there are two pathways. One is the so-called Toll receptor signaling pathway. Another one is called IMD pathway. Then Toll pathway is really dedicated to this response to gram-positive bacteria and fungi. And this pathway involved a very complicated extracellular signaling events, which leads to the activation of a ligand called spatule. And the spatule, once it's activated, can then bind to the Toll receptor, and the Toll receptor then initiate a signaling cascade, which involves several adapter proteins, such as MyD8 and a kinase called PELI. And it's believed that this activation of PELI then leads to the phosphorylation of the Drosophila I-kappa B homolog called cactus. And the phosphorylation of cactus then leads to this degradation, and that liberates the Drosophila NF-kappa B, either dorsal or the diff or dorsal-related immune factor. And then this NF-kappa B can then go to the nuclei to activate downstream targets. And one of the hallmark of the Toll pathway is an antimicrobial peptide called drosomycin. In contrast, the IMD pathway is used to sense the gram-negative bacteria, and that involves a different kind of NF-kappa B molecule. And at the end result of this activation, it leads to a different kind of antimicrobial peptide. In this particular case, one of the hallmark is something called diphtheramycin. So the drosomycin and diphtheramycin can be considered distinct reporters for the Toll versus IMD pathway. All right. So what Bo has really discovered is that And there's an intimate link between the Hippo pathway and specifically to the toll receptor signaling. But unlike the canonical toll receptor signaling pathway, the Hippo pathway is really used to suppress the transcription instead of the protein stability of the cactus protein. Perhaps more importantly, what he discovered that, and the Hippo pathway not only regulates the cactus protein transcription, the Hippo pathway itself actually regulated by the toll receptor. So the story really started when Bo came to my lab, and I told you he wanted to study a Hippo pathway in adult physiology. So what he did was simply take a Galfo driver, which is specifically expressed adult fat body, and knock down core component of the Hippo pathway, these tumor suppressors such as Hippo or Wurtz, or overactivating the activator, Yorkie. And then he subjected these flies to bacterial infection or fungi infection. I show here two examples of gram-positive bacteria, S. aureus and E. facilis. You can see that while Y-type flies, and they are resistant to infection, and these transgenic flies, either RNAi, the tumor suppressors, or overexpressing Yorkie, they are more sensitive. And this phenotype is actually mimicking what happened if you actually knock down the toll receptor. And what is interesting is that if you subject the same flies to gram-negative bacterial infection, they are completely normal. Okay, there's no defect. On the other hand, if you look at a knockdown of a canonical IMD pathway component, such as TAC1, these flies are very sensitive. So consistent with this idea that at the level of bacterial insensitivity, he actually looked at the expression of the specific readout of these two pathways, drosophila versus diphtheria. You can see that, again, we actually, many of this data I'm going to show later on, we use clonal analysis in which you can generate single cells in the fat body, which is the fly immune organ, and ask question, what happened to downstream gene expression or signaling in the intact tissue? So you can see here, this is a single cell, which is missing the Wurtz tumor suppressor. And you can see that, and these flies have been challenged with the gram-positive bacteria or gram-negative bacteria. While the expression of drosophila, I hope you can appreciate, in a single cell that's missing the Wurtz gene product, the expression of drosophila is dramatically reduced, okay? On the other hand, if you look at the expression of diphtheria, which is the readout for the IMD pathway, again, same thing in a Wurtz mutant single cell, the expression is not changed. So this is consistent with the specific sensitivity of these flies to gram-positive, but not to the gram-negative bacteria. In order to trace the molecular defect, right, we know there's something happening upstream of drosophila expression, but how, at which step, and there's a defect. To make a long story short, what Bo discovered was that actually the Hippo signaling pathway and converge on a particular component of the canonical toll pathway, which is cactus, and in particular regulate the transcription of cactus. So I will show you a few slides to demonstrate that. So first, when he looked at the mosaic clones of fat body missing, for example, single cells, which is missing either Hippo or Salvador or Wurtz or MATS gene product, here labeled by the lack of GFP, I hope you can appreciate that in cells with missing these tumor suppressors, they all have a very strong elevation of cactus expression in a cell-autonomous manner. And similarly, you can reproduce the same phenotype by making single cells which overproduce the Yorkie gene product. Right? These cells are positively marked by GFP, and they cell-autonomously express a much elevated level of cactus. And importantly, this phenotype is completely dependent on scallop, which is a partner of Yorkie in transcriptional regulation. Right? So if you make a cell so that overexpress Yorkie but simultaneously mutant for scallop, these cells no longer upregulate cactus. And conversely, if you look at single cells which are missing the Yorkie gene product, okay, here is labeled negatively by GFP, you can see that these cells have much lower expression of cactus. So this actually demonstrates that there's a very, there's a cell-autonomous requirement for the Hippo pathway regulating the transcription of cactus. And so he went on to actually identify the specific element within the cactus locus and regulatory region that's responsible for this transcriptional recognition, which I won't get into detail. But what is striking that he was able to identify a single site that is actually responsible for this recognition. And if you mutate this site, and this transcription can make a laxity reporter, and the mutant version of this reporter can no longer respond to Yorkie overexpression, while the wild-type version, you can see that these are clones of overexpressed Yorkie gene product, and they can turn on these reporters in a semi-autonomous manner, but the mutant version of the reporter is no longer responsive to Yorkie overexpression. All right. So really, what I'm trying to, what he has shown at this point is that the Hippo pathway somehow regulated the transcription of cactus, and by through the Yorkie scallop complex. So the idea is that these flies, when you knock down Hippo, for example, or overexpress Yorkie, they are sensitive to bacteria because somehow they have a high expression of I-kappa B, right, which suppress NF-kappa B signaling, leading to a defect in antimicrobial peptide expression. So if that is the case, you might expect that if you were able to lower the expression or activity of cactus in such a genetic background, you should be able to rescue such an immune-deficient phenotype. So indeed, that's the case. If you take, for example, flies which overexpress Yorkie in the fat body, but you also knock down the expression of cactus, while the overexpression of Yorkie alone leads to a sensitivity, you can completely rescue that phenotype by simultaneously knocking down cactus expression. And you can show this similarly with a different assay in which you can simply measure the bacterial load in such flies. And because normally, if a fly can mount a normal antimicrobial response, the bacterial load in the fly will be very low. However, if you have a defect, the bacterial load in the fly will be very high. However, if you also RNA knock down cactus, you can rescue that phenotype. And I want to draw your attention that these plots are plotted on a log 10 scale. So these are hundreds of folds of difference in terms of bacterial load. All right. So far I've told you that there's a transcriptional regulation by which the Hippo pathway through Yorkie and scallop regulate cactus transcription. So then you might ask, what's the relevance of this kind of genetic control? Is the Hippo pathway there as a constitutive break of cactus transcription, or actually could the Hippo pathway itself being part of the network actually does respond to bacterial infection? So really, the first clue to this idea that the Hippo pathway itself is actually regulated by bacterial infection came from this very simple experiment, which I think is actually a very powerful experiment, in which Bo actually simply look at what happened to Yorkie subcellular localization upon bacterial infection in intact fly fat bodies. So I hope you can appreciate that. Actually, what's interesting, in the wild-type fat body, most of the Yorkie protein is concentrated in the cell nucleus, okay? So the green dye is actually the nuclear dye. You can see that most of Yorkie in the nucleus in the fat body. However, after S. aureus, which is a gram-positive bacteria after S. aureus infection, Yorkie start to be delocalized from the nucleus, and by 2 or 4 hours after infection, you can see that actually most of the Yorkie is now in the cytoplasm. And actually the same phenotype can be recapitulated using a fly immune cell, the macrophage-like cell line, in which you can actually simply treat these cells with gram-positive bacteria or proteoglycan from gram-positive bacteria. You can see that within half an hour, the hypophosphorylation or Yorkie phosphorylation is dramatically stimulated. That's mimicking the temporal response we see in vivo, in which within half an hour, you start to see a Yorkie subcellular localization change, where it goes from the nucleus to the cytoplasm. I should mention that while there are many upstream regulators that have been implicated to regulate the Hippo core kinase cascade from many genetic studies, I should say that most of the studies are static inference based on the loss of function of a certain gene product and how it affects the output of the pathway, and without really knowing the temporal scale by which a particular signal may trigger Hippo signaling in a very short time window. So what we are showing here is that actually gram-positive bacteria can very acutely activate Hippo signaling in intact tissue. So I believe this is actually one of the very first evidence that you can actually have a signal that actually can activate the Hippo signaling in intact tissue in any biological system. So in any case, so this is actually exciting because we can see that for the first time we can actually find that the Hippo signaling can be activated in intact tissue in such a very quick temporal scale. And so this is birth of question, how does that happen? And because we know that the toll receptor plays such an essential role in responding to gram-positive bacteria. And so we hypothesize that maybe the toll receptor itself might be somehow involved in this activation of Hippo signaling. And so to test this hypothesis, again, Bo used a genetic mosaics in which you can make single cells in intact fat body and to overexpress the toll receptor. So in the absence of any gram-positive bacterial infection, he can see that, unlike the normal fat body cell where the Yorkie is in the nuclei, in cells which you actually simply overexpress toll receptor without bacterial infection even, you can see that the Yorkie gene product goes to the cytoplasm. It's no longer concentrating into nucleus. That suggests that the toll receptor is sufficient to activate Hippo signaling in vivo. And he can show similar data in these cell culture-based studies where he can simply activate, overexpress the activated toll receptor or the Pele protein kinase, which is one of the downstream effector of the canonical toll receptor signaling. You can see that both of them can activate the Hippo phosphorylation as well as the Yorkie phosphorylation, again, hallmarks of Hippo signaling. So those are actually, we can now draw an arrow between toll receptor and Pele to the Hippo pathway, and based on this gain of function assay. But perhaps more importantly, are these proteins actually required for the activation of Hippo signaling by bacteria in vivo? So to answer these questions, again, we can turn to the canonical, the classic way we do this kind of analysis, which is make a mosaic. And as a proof of principle, you can see here that if you make a single cell which is missing the Hippo gene product, right, marked negatively by GFP, you can see that upon bacterial infection, or in the Y-type cells, Yorkie translocate to the cytoplasm, no longer concentrated in the nucleus. In the cell which is missing Hippo gene product, it stays in the nucleus, okay? So as if the pathway is not activated. So suggesting that Hippo, of course, is required for this Yorkie translocation to the cytoplasm. And we can do the same experiment for Pele, which is the kinase upstream, and downstream of the toll receptor. I hope you can appreciate that in the Pele mutant clone, and the Yorkie stay in the nucleus. It doesn't translocate to the cytoplasm, right? On the other hand, if you simply overexpress cactus gene product, and in these fat body cell, you do not see a change of Yorkie translocation, suggesting that, however, the toll receptor pathway talk to the Hippo pathway, it has to be somewhere between the cactus protein and the Pele and protein kinase. So, and he also went on to test the other component of the toll receptor signaling pathway, including the ligand spatial, there's toll receptor itself, and the adaptor protein MyD88. And so here, just to remind you, in a Y-type genetic background, if you infect with bacteria within several hours, within actually 2 hours, the protein Yorkie translocated to the cytoplasm, you no longer see the nuclear concentration. And however, in the either toll receptor mutant or the spatial mutant background or in the MyD88 background, you no longer see this translocation. So after several hours after infection, Yorkie remain concentrated in the nuclei, right? So suggesting that all these proteins, which are upstream of the Pele protein, they are all required for bacteria-induced cytoplasmic translocation of Yorkie. So how does this happen? because we know that actually PEDI is required, and all these protein upstream are also required for this translocation. So initially we thought maybe because PEDI is a protein kinase, actually for a long time people have been trying to show that actually PEDI can directly phosphorylate cactus, and without much success. And so we thought maybe actually PEDI can be directly phosphorylating Hippo and somehow activate Hippo. And it turned out not to be the case. So and to make a long story short, what Bo was able to show that is that PEDI can activate Hippo in an indirect mechanism by directly phosphorylating an inhibitor of Hippo, which is a specific type of PP2A complex called the STRIPAC, which was initially discovered by Nick Tapan's lab as a negative regulator of Hippo. So in particular, one of the subunits of this PP2A complex is called the CKA, turned out to be a direct substrate of PEDI protein. And so I should show you some in vitro data to demonstrate that actually PEDI can directly phosphorylate CKA in vitro, but not a kinase dead version of the PEDI protein kinase. But perhaps even more interesting, once you treat the cells, that means immune cells, with the bacteria or proteoglycan from this gram-positive bacteria, CKA protein is quickly degraded within about half an hour. So the protein actually disappeared. And probably more importantly, we can show you in vivo, and actually CKA is absolutely required for this translocation. And for example, if you already showed you in the normal genetic background, right, most of your gene is in the nuclei. However, if you make mutant clones of the CKA subunit of this PP2A complex, you can see that your key is now translocated to the cytoplasm. It's no longer concentrating in the nuclei. And perhaps more importantly, if you actually look at clones which overproduce the PEDI gene product, right? Because PEDI, I already told you, PEDI would degrade, phosphorylate CKA and degrade CKA. You can see that in these cells, they have very low level of CKA protein in the PEDI overexpressing cells. And conversely, if you RNAi PEDI, reduce the activity of PEDI, and you can see that the expression of CKA is very, very high in a cell-autonomous manner. So this actually demonstrates actually PEDI can directly phosphorylate this particular protein CKA, and that leads to the inactivation of PP2A complex, and then that leads to the activation of Hippo. All right? So in conclusion, what I've told you today is that the Hippo signaling pathway is an intimate link between the Hippo pathway and the Toll receptor signaling. And this pathway really regulates a key regulator of the immune immunity, which is cactus, through a transcriptional mechanism. And we believe that downstream of the Toll receptor signaling, there are two branches. One is through the canonical pathway that leads to the cactus degradation in a post-transcriptional mechanism. But meanwhile, there's also a transcriptional mechanism by which Toll receptor will inactivate the transcription of cactus. And both this transcription and post-transcriptional recognition are required for full-blown antibacterial response in Drosophila. So finally, I'd like to advertise that Bo is actually now on the job market. If you see his application, please read his application. I hope you will get a good job. Thank you. All right. Questions?
Great talk. Do you see a problem in innate immunity when you have when you remove scalloped or knock down scalloped?
You mean which one, cactus?
If you see problems when you remove scalloped, not yolky, scalloped.
Scallop. We haven't done that experiment for a very simple reason. So in the imaginal disk, it turns out scallop does not have a phenotype. And so it's has been one of the conundrum for the field for a long, long time. And so the idea being that if you remove the transcription factor, which normally function with your key, right? So you get a de-repression. In another way, you actually get a much weaker phenotype than removing the co-activated Yorkie. When you remove co-activated Yorkie, scallop will engage active repression to repress those targets, which normally can be active by Yorkie. So when you remove scallop, you actually don't get as strong a phenotype as Yorkie. So in many times, actually, in the imaginal disc, you get no phenotype at all. So the reason we can link scallop to Yorkie is because scallop Yorkie double mutant can completely rescue the Yorkie loss of function phenotype. So in other words, that's why we haven't really tested scallop. My prediction would be scallop perhaps will have a very mild or even no phenotype in the immune system because of this default repression mechanism.
Okay.
Right?
Yeah. Hi.
Hi.
I wanted to know if this system is regulating as well Hippo pathway outside of the fat body in the imaginal disc or in other places.
Yeah, that's a great question. I think what we like to think is that this link between Toll and the Hippo is not restricted to the fat body. And I think there's lots of hints about both the Toll and the Hippo being involved in other contexts, such as, for example, cell competition, work from Nora Johnson and many others, and both implicate both pathways in cell competition and other work from in the early embryos involved in convergent extension. So I think this link is not just restricted to the fat body, but we haven't really get into the detail of how these pathways work together in, let's say, imaginal disc. Yeah, but I think that's a very interesting direction to look at.
Thank you.
Yeah.
So you showed really beautifully the change in subcellular localization, for example, in the Hippo and the Pele clones after the Toll pathway activation. And I'm wondering, have you looked at changes in Yorkie levels, if you've seen any differences in the actual total level of Yorkie upon Toll pathway stimulation, as opposed to just a change in the localization?
Yeah, we haven't really looked at it much in vivo, but in the cell culture model, where you can actually infect these MBN2 cell with heat inactive gram-positive bacteria, in those models, we can see phosphorylation, of course, is going way up. We don't see degradation of Yorkie or something like that. So I think that is, generally speaking, in fly, we don't see a degradation of Yorkie, which is very different from the mammalian system, where upon activation of Hippo, the YAP is not only changed localization, it's also degraded. And so there's actually a molecular explanation for why that's the case, because the fly sequence is different from the mammalian sequence, where it's missing a phosphodiagram. Yeah.
Thank you.
Yeah, thank you very much for attending. And one last applause for everyone. That was actually a great session. Thank you very much. And don't forget the posters across the way. Have a lovely lunch.